Hi everyone, my name is Negin Valizadegan. I am a statistical analyst at the High Performance Computing and Biology Center at the University of Illinois. Here I'll be talking about part of my PhD dissertation project on blood microbiome compositions in humans and captive non-human primates and their variations in sensitivity to bacterial molecules. Variations in microbial histories and exposure to microbial molecules have affected primate immune responses. One example of that is sensitivity to a molecule called LPS, so short for lipopolysaccharide, which is a molecule in the cell wall of gram-negative bacteria. The minimum doses of LPS that's needed for inducing a response in humans is much lower than closely related species. LPS can induce a mechanism in the host called immune tolerance. This mechanism actually was discovered when a scientist called Paul Beeson was looking at the effects of E. coli on rabbits in 1947. And he found out that rabbits give reduced responses to vaccine strains of E. coli upon continuous exposure. Basically, he wasn't seeing a fever response in the rabbits anymore after a couple of injections. And this reduced response due to continuous exposure to a stimuli can happen with other bacterial molecules, such as peptidoglycans as well. And it's something that is observed with commensal microbiome and can affect variations in immune responses across species. Blood is a stringent environment that contains hundreds of thousands of professional immune cells that are working to prevent infection. So the common perception is that blood is sterile in healthy individuals. However, there are more and more studies that are finding the presence of bacterial DNA and a lot of bacteria in the blood of healthy humans. But we really don't have a lot of information about non-human primates, blood microbiome, and how variations in microbial communities in the blood can lead to disease susceptibilities. So the central question here is, are primate blood microbial compositions linked to their immune phenotypes? This study has two aims. First is to characterize and compare the blood microbiome across 4 primate species from basically one species from each primate clade. So we have human, rhesus macaque, common marmoset, and ringtail lemur. And the second goal is to get a proof of life. So looking at the levels of lipopolysaccharide in the blood and measure them. And lipopolysaccharide has a very short half-life. So we do expect that we see some levels of live bacterial activity if there is LPS in the blood, circulating in the blood. And the second method we used was culturing blood for bacteria. In order to identify the microbial information, we extracted the DNA from blood. We amplified the V3, V4 of 16S rRNA and sequenced it using the MiSeq platform. A tool called QIIME2 was used to analyze the bacterial information for diversity and tax analysis. And for LPS measurement, the pyrogene recombinant factor C assay was used, which has a broad range of detection of LPS from 0.005 to 5 EU per ml. So the LPS measurements actually showed that we do see an alignment with species sensitivity information. So the box plot on the left shows that we have much lower doses in human, and then it gets a little bit higher in common marmoset, a little bit higher in macaque, and a little bit higher in ringtail lemur. Looking at the actual LPS doses, like a minimum dose of LPS that can stimulate a response, we do see a similar trend in most of the species We do not actually have a lot of information about sensitivity of lemurs, but for the rest of the species, these align very well together. For blood microbiome study, we had a total read count of 6,604,000 reads and a sampling depth of 12,335. Looking at the beta diversity analysis, so two of the metrics that are shown here are weighted and unweighted unifract, which is based on the phylogenetic diversity of these microbes and how distant they are phylogenetically across these primate species. And we do observe a significant difference between the species in terms of both of these matrices. And looking a little bit more closely on at these plots. So I'm zooming in a little bit on the unweighted unifract, which shows a little bit of better separation. Axis 1 is separating haplorhines from strepsirhines, and axis 2 is separating human from non-primates. Axis 3 seems to be separating macaque from everyone else, and interestingly, macaque is the only herbivorous species among these. And then looking at the actual statistical analysis, phylogeny explains 47% of weighted and 38% of unweighted variation. And the pairwise comparison shows that all of these species are different from each other. Looking more closely at what taxa are significantly different, we do see, first of all, a lot of unclassified microbes. We do see a lot of Propionibacterium, which was observed in previous studies as well, in human blood microbiome studies. We have a lot of Proteobacteria in human compared to other species. And we do see common gut microbes and pathogens that are observed here, including Enterobacteriaceae, Lactinospiraceae, Ruminococcaceae, Calensula, for example. Calensula is known to increase gut permeability by reducing the expression of tight junction proteins in epithelial cells. So we were curious to see what the potential sources of these microbes were. And I used a tool called FEAST that allows us to detect the sources of microbiome. And looking at multiple different body sites, you see that the fecal microbiome is explaining a much higher proportion of this, the blood microbiome. And then looking a little more closely at what's shared, or the core microbiome. So this is what's shared across 20% of samples in blood and feces. We do see that most of these, everything except for the unknowns, are much higher in feces, and potentially explaining that we might have a translocation from the gut to the blood. We have a lot more unknowns in blood, which aligns with the fact that blood is understudied, and the databases we have might not be not contain a lot of information about blood microbiome. And then two microbes from the Proteobacteria phylum. So it's a major gram-negative bacteria that was observed much higher in humans. Are also seen here among the shared microbes between feces and blood. And blood actually has a lot of the Enterobacteriaceae. Looking at the phylum level, we observe differences in relative abundance of bacterial phyla, especially Proteobacteria and Ectobacteria as the two most dominant blood bacterial taxa. And splitting these microbes into gram-negative and gram-positive. Gram-negative is shown in the red bracket, and positive in the green. We do have a lot higher proportion of gram-negatives in more sensitive species, and also a lot more fat consumption. And previous studies have linked higher fat consumption to higher amount of gram-negative bacteria and bacterial translocation from the gut to the blood. So to summarize that information, we found that blood microbiome is varied across primates, both in terms of diversity and bacterial taxa, including relative abundances of gram-negative versus gram-positive bacteria. And with that, I'd like to thank my PhD committee members for the intellectual contribution and support, the sample and funding sources, and friends and colleagues. I thank you for listening to this presentation.